Good morning children, welcome for today's biology class. So today we are going to study the chapter Biomolecules. So we are going to cover how to analyze chemical composition, primary and secondary metabolites, and among biomacromolecules we are going to focus upon lipids. First let us study how to analyze chemical composition. The living cells are composed of both organic and inorganic compounds. So how can you find out? So for that purpose we are doing some chemical analysis. For organic compounds we do one type of analysis. For inorganic compounds we do other type of analysis. For organic compounds, the living tissue what to be analyzed, along with that trichloroacetic acid, that is mixed and it is grind together. You have to grind together to make in the form of slurry. Okay, like mixture, it has to be ground together. Then later the slurry it has to be filtered. So when you filter, you will get two fractions. So one will be filtrate, it will be watery, that is acid soluble, and other will be retentate. So that will be acid insoluble. Okay, so like that the two fractions among these two, no, so that acid soluble pool, the acid soluble pool, no, it contains all the organic compounds. The all organic compounds are present in acid soluble pool, that is filtrate, that you will get. Okay, next for inorganic compounds. For inorganic compounds, the sample tissue should be burnt. Okay, it has to be burnt dry. So to obtain ash. So then later different kinds of inorganic compounds are identified in that. Next one, let us study regarding the type of biomolecules. There are two types of molecules are there. So one is micromolecule, other is macromolecules. Micromolecules are known as monomers. Macromolecules are known as polymers. Micromolecules are simple form. Macromolecules are complex form. Okay, so now let us study regarding primary and secondary metabolites. You know what is metabolism? The biochemical reactions taking place within our body, you call that as metabolism. The metabolites, so they are no resultant of metabolism. So these are biomolecules seen in living cells. Metabolites are the biomolecules seen in living cells. So primary metabolites are, so when normal physiological process, so when it is produced, so that you call that as primary metabolites. It can be amino acids, nitrogenous bases, proteins and nucleic acids. Secondary metabolites, so they are the product of certain metabolic pathways from primary metabolites. Okay, it is derived from primary metabolites. So example it has been given. So 5 examples, it can be asked in 5 mark questions also. Sometimes by giving the example they can ask which category of metabolite, secondary metabolite they belong to, and they can identify whether it is, you have to identify whether it is a primary or secondary metabolite, like that. So pigments, anthocyanin pigment, carotenoids, you know, beetroot it contains anthocyanin pigment, and carrot you can see carotenoids, then drugs like vinblastine, curcumin, okay, then alkaloids, morphine, codines, then essential oil, that is lemongrass oil, okay, then polymeric compounds, they are rubber, gum, cellulose, resins, so all are coming under secondary metabolites. These are the during metabolic pathways, no, it is obtained from primary metabolites, that's why you call that the secondary metabolite. Then biomacromolecule. What is biomacromolecule? This is a molecule and the weight will be greater than 1000 Dalton and it is found in acid insoluble fraction. Okay, so it can be polysaccharides, nucleic acids, proteins, lipids, all are coming under this category. So biomacromolecules, so they are of 4 kinds. Proteins, so proteins, it is a polymer composed of amino acids, and carbohydrate, no, the simple sugar, so it is no simple sugar, it is made into polymer, so means it will be converted into carbohydrate, it is a polysaccharide. Okay, carbohydrates are various types, it can be monosaccharide, disaccharides or polysaccharide, like that it can be. And lipids, so lipid, no, it is a fat, oil, wax, okay, or a steroid. And nucleic acid, so you know that nucleic acid, there is DNA, RNA, okay, so it is a polymer of sugar an organic base and phosphoric acid. So we will study detailedly, children, in this chapter everything. So first let us study regarding the average composition of cells. So what are the components, how much it is present. So water it is about 70 to 90% it can be there. So proteins about 10 to 15%, carbohydrate about 3%, lipids about 2%, nucleic acid about 5 to 7%, and ions remaining it can be 1%. So when you see a cell, approximately this type of components will be there. First, let us study regarding amino acids. Amino acids are micromolecules. So these are organic compounds containing an amino group. So that's why the name amino acid and then acid group. Okay, so that's why the name we are giving amino acids. So actually, so both are present on the same carbon. So that is, the carbon is alpha carbon. So that's why amino acids, they are known as alpha amino acids. They are substituted by methanes. Okay, so there are 4 constituent groups are occupying 4 valency positions. So it will be hydrogen, carboxyl group, amino group, and R group. It is variegated, it is variable group. See here, it is the amino acid, simple amino acid, glycine, carboxylic acid, and you can see hydrogen on one side and amino group, and this is the R group. What is given in box, it is R group. You can see in all amino acids, glycine, alanine, serine. So everything is same, only the box portion it is varying. In glycine, it is hydrogen is present. In alanine, you can see that is methyl group, and in serine you can see the hydroxy methyl group. So only in the R group, the amino acid will be varying. So based on the nature of R group, there are many amino acids are there. But anyhow, total amino acids are about 20 amino acids are there. But in a protein, so the 20 amino acids in various combination, they form proteins. Okay, but type of amino acids are 20. So this one is studied, children. So the R group, it vary from amino acid to amino acid. So next, let us study regarding lipids. So lipids are insoluble in water. So they are simple fatty acids. A fatty acid has carboxyl group attached to a R group. Okay, so the R group, it could be methyl group or ethyl group or higher number of CH2 groups. It can vary from 1 carbon to 19 carbons. So for example, when you see the palmitic acid, it has 16 carbons. Okay, including the carboxyl carbon. So when you observe arachnidic acid, okay, it has 20 carbons. Like that, it vary from fatty acid to fatty acid. So when you see the fatty acids, it can be 2 types. It is saturated or unsaturated. Saturated means without double bond. Unsaturated means they have 1 or more double bonds. Okay, so when it is that, you call it as unsaturated. So then another simple lipid is glycerol. The glycerol is trihydroxypropane. So many lipids, they have both glycerol and fatty acids. Then accordingly, it can be monoglycerides or diglycerides or triglyceride. Okay, the lipids, so when you observe, they are also, you call them as fats or oils based on the melting point. So in plants, what do you get? You call that as oil. Normally fats we get from the plants, we get oils, and animals we get the fats. So oils have lower melting points. For example, gingerly oil, groundnut oil, like that we give oils. Oils, they have low melting point and they remain as oil and as liquid even in winters itself. But when you observe, coconut oil is exception. They have a special octanoic acid. Because of that, it gets solidified during winter. When you see the other normal plant oils, all will be, so they will be liquid during even at low temperature also. But when you observe animal fats, they are solid at room temperature. Okay, you call that as plant oils and animal fats. So because in the case of animals, they have the fatty acid present is saturated fatty acid. Okay, so here we studied saturated fatty acid. In plants, they have unsaturated, and unsaturated can become saturated. When you make a vanaspati, the dalda, so when you make like that, so it is by means of the opening of bonds between this one, the unsaturated will become saturated. Okay, then it will become the melting point. So in that case, what happens? They will be, because of that, they will be solid. even at room temperature also they will remain as solid. It modifies the melting point. So oil, they have lower melting point, but the animal fats, no, they have higher melting point. That's why they are solid at room temperature. Then phospholipids. The phospholipids, they have phosphorus, okay, or phosphorylated. They have no phosphorus in the lipids, that is conjugated lipids. So that's why you call them as phospholipids. And example, so lecithin, okay, there is one example for that. They are mainly seen in, no, these phospholipids are seen in cell membrane. So some tissues, especially the neural tissues, they have lipids, and they are, that is no neurons, they have no lipids, and they are of more complex structure. Okay children, these are different structural formula it has been given. So you can remember for palmitic acid and for glycerol. Okay, then the other thing, no, triglyceride, just you can go through, no need to remember, because no, it is not much needed. But when they ask this two, you can remember glycerol and palmitic acid, and it is no triglyceride. So can you see R1, R2, R3, okay, they all are fatty acids. That's why we call it as triglycerides. It is phospholipid. Example, lecithin, it is cholesterol. Okay, so we have studied structural formula for various lipids also, and 3 amino acids also, and 3 amino acids, and the first 2 lipids, no, whatever we studied, this one you can remember, this glycerol and palmitic acid you can remember. So others just you can go through. Okay children, so today we are concluding up to this. We will meet in the next class. Thank you children.